Assalamualaikum and hi everyone. I am Nurisma binti Tajul Kahar. Today I'll be presenting on an antibiotic called Novobiocin. So first let's begin with some background information on Novobiocin. Novobiocin is an antibiotic derived from the actinomycete Streptomyces nevis that is usually isolated from the soil. It shares a chemical structure similar to coumarin, which is a colorless crystalline compound with a sweet odor and bitter taste, commonly found in many plants. Novobiocin sodium was first approved for medical use in September 1964, primarily to treat serious infections caused by susceptible Staphylococcus aureus. However, in 2009, the Food and Drug Administration withdrew it from the market due to concerns regarding its safety and effectiveness. As of today, Novobiocin is no longer used in human medicine, but it is still approved for veterinary use in Taiwan. Now, moving on to the general characteristic of Novobiocin. It belongs to the aminocoumarin class of antibiotics. It has a 95% protein binding capability. It is insoluble in water and has a molecular weight of 612.6 grams per mole. Novobiocin is generally more effective against gram-positive bacteria compared to gram-negative one. In veterinary medicine, Novobiocin is often combined with other agents to treat bovine mastitis. It is an inflammation of the mammary gland caused by pathogens such as Staphylococcus aureus, Streptococcus agalactiae, E. coli, and Enterococcus species. It is also used in laboratory settings to differentiate between coagulase-negative Staphylococcus species. It can also be used as a selective culture media supplement to inhibit gram-positive organisms for the microbiological analysis of food, animal feed, water, and environmental samples. Let's now look at the mechanism of action. Novobiocin works by binding to the ATP binding site of the ATPase subunit in DNA gyrase, an essential enzyme that introduces negative supercoils into bacterial DNA. This binding inhibits DNA gyrase activity, which interferes with DNA replication and transcription. Additionally, Novobiocin can activate ATPases involved in lipopolysaccharide transport, altering cell membrane permeability and allowing the antibiotic to enter and disrupt bacterial functions. Differences in the structure of cell membrane between gram-positive and gram-negative results in different responses against Novobiocin. Gram-negative bacteria are naturally resistant to many antibiotics due to the presence of the outer membrane, a unique asymmetry bilayer with phospholipids in the inner leaflet and lipopolysaccharide in the outer leaflet. A key example of a bacterium susceptible to Novobiocin is Staphylococcus aureus. Next, let's discuss resistant mechanisms. Resistance to Novobiocin typically arises from point mutations in the GYR beta gene, which encodes the B subunits of DNA gyrase, the main target of Novobiocin. These mutations prevent the antibiotic from binding effectively to its target, allowing the DNA replication process to continue normally. An example of a Novobiocin-resistant bacteria is Staphylococcus saprophyticus. Staphylococcus saprophyticus is a common cause of community-acquired urinary tract infections. It is one of the few Staphylococcus coagulase-negative species that are resistant to Novobiocin. Lastly, here are some of the references I used for the poster, and thank you for listening.